Dear student, last time we started our learning process onto a very large and diverse group of microorganisms belongs to the family Enterobacteriaceae, and we were discussing one of the important organisms, which of course is a gram-negative facultative rod that ferment lactose, and we were discussing the Escherichia coli. And we all know that the Escherichia coli has three important antigens, and these important antigens are used for the identification of the organism during epidemiological investigations. Now, we have the O antigen, what we call as the somatic antigen or the cell wall antigens. We have a flagellum antigen, which is designated as H antigen, and we have the K or the capsular antigen. We have more than 150 antigenic variations of the O antigen, over 50 antigenic variations of the flagellum antigens, and about 90 variation of the capsular antigen. So when you see these various combinations, they result almost more than 1,000 subspecies of the Escherichia coli. Now, based on these subspecies, we said that we have one important organism, that is O157H7. This is what is the somatic antigen 157 and flagellum antigen combination, and that was involved in the Escherichia coli associated diarrhea. We now move on to, excuse me. Yes, we discussed that the gastrointestinal disturbances caused by the Escherichia coli, and we will now start on to the urinary tract infections because of the Escherichia coli. Now, the certain O serotypes of Escherichia coli, they preferentially cause the UTI infections, particularly the uropathic Escherichia coli, what is abbreviated as UPEC. It is a stain which is characterized by the presence of pili, and it has, with these pili, it has various adherence proteins, and these proteins, they bind to the specific receptors on the urinary tract epithelial cells. But as we know that all gram-negative organisms, as is in case of Escherichia coli, mostly they are with the fimbriae or the pili. Now these pili, they provide a region of the microorganism with the intestinal mucosa, and after a region, the organism, it then penetrates, it multiplies, it secretes different toxins and enzymes, and that really causes damage to the GI tract. So after entrance, the proteins, the specific receptor, there is binding, and then you see the motility of the Escherichia coli. The Escherichia coli, because of its symmetry and its flagella, its motile, they may aid in the ability to ascend the urethra, even into the bladder, or even the ureter into the kidney. If you look at this this picture over here, you see the motility of the microorganism. When the GI tract is heavily infested, then it can cause, even you see the ascending of the urethra into the bladder and all that. It has been seen that the cranberry juices, it has some component which is known as the flavonoids. They are useful in the prevention of recurrent UTI infections because they block these receptors. They block these receptor sites which are present on the GI tract. So all those individuals who have recurrent E. coli infections in the GI tract, particularly the female, because of their close contact of the urethra with that of the vagina, they are really the main target of this organism. And this, you see, the juices, they can really help in that sense. Now, the other systemic infections which can be had with the Escherichia infection. You see, we have the two structural components in the Escherichia coli. It's a capsule and it's endotoxins. Now, both of these, the capsules and the endotoxins, they contribute towards the vigilance of this microorganism and causes systemic infections rather than the intestinal tract diseases. So the intestinal diseases, these are because of the fimbriae that provide adhesion of this microorganism with the mucosae, whereas the systemic infections are because of the capsules and the endotoxins which are really released by these microorganisms. The capsule, as we all know, the capsule are polysaccharide in nature, and this phagocyte, this capsule, it interferes with the phagocytosis and thereby enhancing the organism's ability to cause infection in the organs. The Escherichia coli certain stains, they cause neonatal meningitis, and these stains, they have a specific capsular type, what we are called as the K1 antigen. So the antigen of the capsule, which has been designated as K1, it is involved in the neonatal meningitis in children. The endotoxins, which are of course the salva lipopolysaccharides, they have a generalized, you see the mode of actions and the generalized signs and symptoms that are really designated in the clinical infection. It causes several features of gram-negative sepsis. Now, what is the outcome of the sepsis? There is a fever, there is a hypotension, and there is disseminated intravascular coagulation. So these signs and symptoms that we can see clinically as because of the production of endotoxin, which is lipopolysaccharide in nature, and this is the generalized characteristic for all the gram-negative rods. Now, some of the individuals, particularly the immunocompromised individuals like HIV, they are predisposed to sepsis because of the Escherichia coli and Klebsiella. And this is due to the loss of the T helper cell 17, which result, you see, they help in the sense of the interleukin 17. And interleukin 17 has the role to play to avoid the sepsis. So all those individuals suffering from some kind of immunodepression or immunosuppression because of the drugs, because of the diseases, because they are really the good target for the systemic infection of Escherichia coli and the Klebsiella, particularly the Klebsiella pneumoniae, which has a huge capsule. Again, you see the capsule, always, whenever there's a capsule forming stains of any microorganism, the capsule, it acts as an antiphagocytic factor and it contributes towards the virulence of the organism. So the organism is equipped enough itself for the production of the disease or initiate disease in the human body because of the endotoxins, because of the fimbriae, because of the capsules. Now, the clinical findings of systemic infections, you see, the organism is the leading cause of community-acquired UTIs, and primarily in the women, because if you look at this diagram, there's a short urethra, and the proximity of the urethra to the anus, it helps in the colonization of the vagina, facilitate ascending infection to the bladder. So the females, they are more prone to UTIs compared to that of the male because of the short extremities, proximity of the anus with that of the vagina, and the vagina is highly contaminated with this organism. The most frequent cause of nosocomial infection, what we call the hospital-acquired UTIs, and this is associated with the indwelling urinary catheters in both the sexes. The cystitis, which is the dysuria, and frequent discomfortable urination, along with that pyelonephritis, is designated with the fever, chills, and the flank pain. So in females, the Escherichia coli associated UTIs are very common, and they are associated with the cystitis and pyelonephritis. There is a high rise of temperature, chills, and even the flank pain. So we have to go for the antimicrobial chemotherapy, regardless of the sensitivity testing. Other findings that one can see with that of this Escherichia coli is the major cause of meningitis and sepsis in neonates. You see, we have another group of microorganisms, which is a group B streptococci, which are responsible for the meningitis and sepsis in neonates. And this is the second most important organism, along with that of the group B streptococci. They frequently associated from the patient with hospitalized acquired sepsis. Arises primarily from the urinary, biliary, or peritoneal infections. Peritonitis is a mixed infection, and it can occur with that of Escherichia coli or other facultative enteric organisms, particularly the anaerobes of the colon flora, the bacteria, the fusiform. These bacteria, the bacteria fragillaris, they, along with that of Escherichia coli, they cause mixed infection, particularly when you see, if you are performing some laparotomy, are you undergoing some of the, are you are having your patient with that of some of the GI or abdominal surgery, these bacterial infections are very common. So the best way to avoid the infection or peritonitis because of the Escherichia coli or peritonitis because of the bacteria or fusiform is to give some pre-medication avoid these infections. Other clinical findings, you see, that based on to the various stains of the Escherichia coli. We all know that we have the enterotoxigenic Escherichia coli, which is termed as EHEC, and it causes usually watery diarrhea. There is no blood. It is self-limiting diarrhea of short duration, and this is what we refer to be as the traveler's diarrhea. So traveler's diarrhea is caused by the enterotoxigenic stains of Escherichia coli. The dysentery like syndrome, which are characterized by the bloody diarrhea, abdominal cramps and fever, this is caused by the enterohemorrhagic Escherichia coli. Enterohemorrhagic Escherichia coli, and we have the enterotoxigenic Escherichia coli, and the symptoms of the enterohemorrhagic Escherichia coli, they are similar to that of the Shigella, because the enterohemorrhagic toxin, it acts the same way if we have the Shigella toxin. The another classical stain of Escherichia coli, that is what we call as the O157H7, it causes bloody diarrhea also. So we have two stains of the Escherichia coli, enterohemorrhagic Escherichia coli and the O157H7 that causes bloody diarrhea. And this, you see, the OH157 infection, it is complicated by the hemolytic uremic syndrome. In this, what we call it, there is the kidney failure, there is hemolytic anemia, and thrombocytopenia. This occurs particularly in the children which are being treated with the fluoroquinolones or other antibiotics that are used for the diarrhea. For this reason, the antibiotic, they should not be used to treat the diarrhea, which is caused by the enterohemorrhagic stains of Escherichia coli, particularly the O157H7. We have to be very careful while prescribing broad spectrum antimicrobial drug, particularly in the infants and children, because they can undergo kidney failure, resulting into the hemolytic anemia and thrombocytopenia, and later on maybe the permanent damage to the kidney due to kidney failures. Now, the lab diagnosis of the gram-negative rods is generalized, as we have seen previously. The organism can be grown onto the, initially we have to use two media. The blood agar is there, and one of the differential selective mediums like the McConaughey agar, the eosin methylene blue agar, the Escherichia coli, they are the lactose fermenters, and they form pink colonies, whereas the lactose non-fermenters or non-lactose colonies are formed by the other organism of the gram-negative rods. On eosin methylene blue agar, the colonies, they have a characteristic green sheen appearance, as we have seen over here. And the Escherichia coli O157H7, it does not ferment sorbitol. And this is an important test or distinction criteria between the other stains of the Escherichia coli and the E. coli O157H7. We perform the biochemical test, what we call as the sorbitol fermentation test. If it is positive, it is Escherichia coli stain O157H7. It is negative, these are the other stains of the Escherichia coli, the other enterotoxigenic or enterohemorrhagic stains of the Escherichia coli. The isolation of the toxigenic stains and pathogenic stains of the patient with diarrhea is not a routine diagnostic procedure, but based on the clinical signs and symptoms and the history, we can really start with the treatment. But if you see sometimes the resistance stains are there, you will have to go for the antimicrobial sensitivity testing in the laboratory. The treatment for these anaerobic, facultative anaerobic gram-negative organism, it depends upon the disease and the resistance pattern of the specific isolates. Normally, you see the 1 to 3 days treatment with oral ampicillin or with that of the trimethoprim, sulfamethoxazole, which are available in the combination of Septron or Bactrim. We can use that. The sepsis, we need parenteral antibiotic treatment. Particularly, we need the third generation cephalosporin, ceftriaxone, ceftexim, with or without aminoglycoside, gentamicin. Sometimes we need a combination because the sepsis is a fatal disease. It's a life-threatening complication. We'll have to take care of this aggressively. We will have to go for the combination of treatment, cephalosporin with that of the gentamicin. In neonatal meningitis, which is again, you see the combination of ampicillin and fourth generation Piloran is required. The antibiotic therapy, usually, it is not indicated for the routine diarrheal diseases caused by the Escherichia coli. And the administration of the sulfamethoxazole, trimethoprim, loperamide, like the Imodium, they may shorten the duration of the symptoms, but we hardly need antimicrobial chemotherapy for this purpose. That was all about the Escherichia coli. Now we move on to the other important genera, which is again a gram-negative rod. It's a facultative anaerobe. It makes a capsule, and it causes diseases like enterocolitis. It causes enteric fever, what we called as the typhoid fever. It causes septicemia with the metastatic infections, particularly the organism lodge into the bones and causes osteomyelitis. And this is the most common cause of the bacterial enterocolitis. We have the organism, what we call as the Salmonella typhi, Salmonella paratyphi A, B, C, and we have the non-typhoidal Salmonella that belongs to this group. Again, we have to concentrate on to the different important properties of this microorganism. It's a gram-negative rod, it's a non-lactose fermenters, and this produces hydrogen peroxide, hydrogen sulfide, H2S. H2S is produced by the fermentation of the iron. The ferrous sulfate is deoxidized into ferric sulfate, and it results in the beckoning of the medium, as we have seen in case of the triple sugar iron medium that we discussed in the very early stage. Again, you see the antigens are very important in this microorganism. If you look at the entire medical microbiology, the somatic antigen, flagellar antigen, and capsular antigen, they are very, very important in case of Escherichia salmonella. Now here we have the somatic or flagellar and the cerebral antigen. We have the flagellar antigen, we have the capsular antigen. Now the capsule in case of Salmonella typhi, which is a real human strain of the Salmonella genus, it is virulent. That is why it has been always designated as the virulence capsule. And we designate that VI, the VI virulent capsule of Salmonella typhi. Now the O antigens, we can divide the total Salmonella into different O antigen groups starting from A to I. The flagellar antigen, it occurs in two phases, what we call as the phase 1 and phase 2. It's a biphasic microorganism. Virulence antigen is a capsular polysaccharide, and the virulence antigen is used for the serotyping. So again, you see, we have a huge group of microorganisms, huge group of Salmonella that is based on to the antigenic variation of cell wall antigen, the flagellar antigen mostly, and to some extent the capsular antigens. Now, there are three methods for the nomenclature or for the broader study of the Salmonella. Now, there were two scientists who have really contributed their lives to the nomenclature designations of this microorganism. One of them is the Ewing. It has a three species concept. The Ewing says that we have broadly three main species in the genus Salmonella. The Salmonella typhi, which is exclusively an human strain. You will not find Salmonella typhi in any species of the animals. Not in arthropods, not in animals like buffaloes, cattle, or in swines, or in goat, or something like that. It is exclusively human-oriented strains. All humans, they will have typhoid with the help of Salmonella typhi, not with other organisms. Then the second most important organism that he designated was the Salmonella cholerazuus. It causes disease in the swines. This is again host-specific. It will hardly found in the humans. The humans only get this cholerazuus infection if they consume or they eat the raw, or undercooked meat of the swines, of the pigs. And then we have another strain which we call as the Salmonella enteritidis. This is found in the humans also. This is found in the animals and the humans. Animals are the main reservoir for this enteritidis, like the sheep, the goat, the cattle, and the poultry. So three exclusive broad categories of the Salmonella genus. According to the even with we have the three species concept: Salmonella typhi exclusively for humans, Salmonella enteritidis for rest of the animals like buffaloes, cows, sheep, and all the, and Salmonella cholerazuus for the swines. Now this was the three species concept of the evening. Now we have another scientist, what we call as the Kaufman and White. It has a different species name to its serotypes. You see, it has the Salmonella Dublin according to Kaufman and White, would be the Salmonella enteritidis serotype Dublin according to the evening. So the evening has the Salmonella enteritidis, But we have the substance, what we call it Salmonella dublin. And Kaufman and White says that instead of saying this the Salmonella enteritidis, they were said Salmonella enteritidis serotype dublin. This means that we have many strains of the Salmonella enteritidis. Enteritidis is not just a single strain. We have hundreds and thousands of strains of the enteritidis. Salmonella chlorozoas is a different. So in case of, you see, it will be a Salmonella enteritidis. See, Salmonella enteritidis serotype chlorozoas, Salmonella enteritidis serotype dublin. So these are the various nomenclatures. You just have to, remember to some extent that how these species are classified. Otherwise, they're not having any clinical significance. Of course, they do have the epidemiological significance, but they don't have the clinical significance. We can also classify a nomenclature. We can on the DNA homologies. Clinically, there are two distinct categories of the Salmonella: the typhoidal species and the non-typhoidal species. This is what you have to keep in mind. The Salmonella are broadly categorized into two: the typhoidal Salmonella and the non-typhoidal Salmonella. The typhoidal species are the Salmonella typhi and Salmonella paratyphi, right? Salmonella typhi and Salmonella paratyphi. Whereas the non-typhoidal species, which causes enterocolitis in humans and metastatic infections such as osteomyelitis, the Salmonella enteritidis, Salmonella chlorozoas. So we will confine our discussion in the medical microbiology just onto the three species concept. Three species concept of evening, we have Salmonella typhi, which is mainly exclusively for the humans. It causes typhoid fever. And we have another strain, what we call the Salmonella paratyphi. It has three subcomponents: paratyphi A, paratyphi B, paratyphi C. So typhoidal strains, non-typhoidal strains. Typhoidal strains are the Salmonella typhi, Salmonella paratyphi. The non-typhoidal strains are the Salmonella chlorozoas, Salmonella enteritidis. They causes enteritidis, causes enterocolitis. And of course, some of the metastatic infections when you see the bacteremia and septicemia develop, and the organism lodge in the bone cause osteomyelitis. Now, the pathogenesis, there are three types of infections which are being caused by the Salmonella organism. Of course, the enteritidis, it causes enterocolitis. The typhi, it causes the typhoid fever and enteric fever. The typhi, and it causes septicemia also. Enterocolitis, you see, there is epithelial, subepithelial lesion of the small and large intestine after penetration of the mucosal cells of lemnopropia. There is inflammation and there is a diarrhea. And of course, when there is a penetration, there is a PMN's leukocyte response limit the infection to the gut and the mesenteric lymph nodes. The Shigella, you see, the dose, the infection dose is very less. It is just 10 cells, but we need about thousands of cells of Salmonella to cause disease. So it's not easily gettable disease. Well, the body is very well tolerated, and but we need a huge number of cells compared to that of the Shigella. The gastric insert, it impairs the host defenses, and gastrectomy and the antacid use, it reduces the infectious dose of the Salmonella organism. So Shigella, they are more virulent, they are more pathogenic compared to the Salmonella because we need a high number of microorganisms of Salmonella enteritidis or Salmonella chlorozoas to cause enterocolitis, compared to that of the enterocolitis caused by Shigella dysentery, where we need just from 10 cells, or it may be from 10 to 100 cells, are enough to cause the disease. The typhoid and the other enteric fevers, the infections begin in the small intestine. Then they enter, they multiply in the phagocytes of the Peyer's patches and spread to the hepatic, splenic, and gallbladder phagocytosis. Bacteremia develop, and the fever is due to the disease of the endotoxins and the survival growth within the phagosome and the phagocytic cell. And even of the gallbladder cancer. Now, one of the main pathogen outcome that we have to keep in mind, that the patient becomes carrier. Once infected or having disease of Salmonella typhi, the patient becomes carrier, and it go on excreting the bacteria and the feces for a long period of time. The organism lodge in the gallbladder, because after we enter the gallbladder, this is perhaps the more important component of the pathogenesis of the Salmonella typhi. The organism lodge into the gallbladder, and this persistent presence of Salmonella typhi in the gallbladder and continuous secretion or excretion of the bacteria and the feces for a long period of time, the individual becomes a source of infection to the other. And there are lapses of typhoid. because most of the antimicrobial drug that we use to treat the typhoid fever, they do not reach in the gallbladder in the required therapeutic concentration. So they do not attain the therapeutic level, and the organism remain there, and it become resistant, and the patient become carrier. So once infected with the typhoid, organism is lodged in the gallbladder, you are the carriers, you can undergo yourself into recurrent of the disease, what we call it the relapses of the typhoid fever, and the other individual can be exposed to this infection from the feces of the contaminant of the patient. Now, there is septicemia is also there in most 5 to 10% of the chronic diseases, particularly because of the sickle cell anemia patient or the cancer or child with enterocolitis. They are more prone to this septicemia development. The organs, they become seedling and leading to the lungs, in the brains, and in the bones. So the common sequelae of these meningitis, the pneumonia, and osteomyelitis, we can face that. Osteomyelitis in children with sickle cell anemia is very common, and metastatic abscess in the previously damaged tissues, most frequently at the site of infarctions. And this is an important cause of vascular graft infection in facultative, anaerobic, gram-negative rods. They are an important cause of the vascular graft infection also. Epidemiology of the salmonella infections, of course, it's by the ingestion of the contaminated food and water. And salmonella typhi is, you see, really transmitted only by the humans to human. A non-typhoid salmonella is like we have the salmonella enteritidis, salmonella colazuas, salmonella typhimurium. They have significant animal and human reservoirs. Poultry meats, egg, they're the most frequent source of infections in human. In Pakistan, you see, studies have been conducted by the several, see the microbiologists, and it's been seen that the poultry, meat, and egg are the main source of infection in of salmonella, particularly the eggs and the poultry. Now, because of the cooking style, we do not get the infection, but the birds, they're the carriers, and we have the huge amount of these microorganisms prevalent in the poultry, meat, and eggs. So be careful. In Pakistan, mostly you see the Salmonella enteritidis, and it has been fast type, the fast type 4. Salmonella enteritidis fast type 4 is the most common cause of enterocolitis, poultry-related or egg-related or bird meat-related enterocolitis in Pakistan. The additional sources can be the dog, pets, turtles, snakes, lizards. You see, name and animals even have the salmonella. Non-typhoid salmonella are very, very common, very prevalent, very common. The clinical findings, the incubation period for this organism is about 12 to 48 hours. Initially, there is nausea, later on enterocolitis develop, vomiting, abdominal pain, and mild to severe diarrhea with or without blood. So we can have the bloody diarrhea, what we call it the dysentery. We can have the normal diarrhea, vomiting, nausea, and abdominal pain. The disease is normally self-limiting after a few days. It goes for some time and then it vanishes. The medical care is not required except in a very, very young and very, very old individual. The immunocompromised individual and the very young children, they require some of the medication. Otherwise, it's self-limiting. Of course, the HIV-infected individual who have the low CD4 count, they are much more prone to the salmonella infections. There is more severe diarrhea. There is more serious metastatic infections. And this is true with all the other microorganisms. AIDS individual in which the CD4 is very low. This is the organism, they have all the chances to get into cause severe diseases and metastatic infections. The Salmonella typhimurium, this is one of the main common cause of enterocolitis in the United States, but in Pakistan, we do have many cases of the Salmonella typhimurium also. Now, the clinical findings with reference to the Salmonella typhi that causes typhoid fever are enteric fevers which are being caused by the Salmonella paratyphi A, B, or C. The Salmonella paratyphi B and C. Typhoid fever is caused by Salmonella typhi. Enteric fevers are caused by Salmonella paratyphi A, B, and C. And the Salmonella paratyphi B and C, they have also been known as Salmonella Scottomilleri or Scomata hickfeldi, Salmonella hickfeldi, and Salmonella scottmilleri. They are the other two names for the paratyphi species belong to the group B and C. There is slow onset of the disease, and of course there is a fever, constipation, no nausea, no diarrhea, no vomiting in case of typhoid, the typhoid strains of the Salmonella. If later on the bacterium develop, there is a high fever, there is tender abdominal, enlarged spleens, rose spot, which are the rose-colored macules, and the leukopenia, anemia, and abnormal LFTs, liver function tests, they're very common. So rose spot is almost the pathognomonic, the Leon's clinical clues. The disease begins to resolve by the third week. Severe complication, particularly the intestinal perforation and hemorrhage, this is very common. So always you see, we have to look into that. There is the swelling and later on perforation and hemorrhages and perforation of the, it's a complication. The complication of typhoid fever is intestinal perforation, which later on, of course, when the perforation, there will be peritonitis and very, fatal complications can arise. Now, this is how the rose spot will look like in a patient of typhoid fever. There are rose-colored macules, particularly on the trunk and on the back, and sometimes you may have on the face. So the characteristic clinical clue is really along with the high fever and splenomegaly and the shift in the LFTs, we can identify or we can make the diagnosis on clinical basis. The clinical findings again continue with that of the enteric fevers. There is 3% chronic carriers, especially with the gallbladder disease and gallstones, right? So if you have the gallbladder disease, you have a gallstone, the organisms are there, you are the carriers, and you are also exposed to recurrent of or the relapses of these typhoid fever. Septicemia most often caused by the cholera zoonose. There is high fever. There is no enterocolitis. Another focus symptoms are associated with the affected organs. Whichever organ is involved, you will have the symptoms like that. Frequently, the bone, lungs, and the meninges. The lab diagnosis of the Salmonella in case of enterocolitis, we isolate from the stool samples. In case of enteric fever, the blood, the bone marrow cultures are positive. We can go for that. For the stool cultures, positive chronic carriers organism, and there's non-lactose fermenters colonies on McConkey agar, as we have seen. They appears, you see the whitish in color, the eosinophilic blue, they're mostly while the golden color colonies. On triple sugar island, there is alkaline slant, acidic pH, mostly with the gas and acidic black color in the butt. Black color in the butt. This production of gas means that the butt may have been thrown upwards to some extent, and there is a reclined slant. Typhi does not form any gas and produce small amount of H2S. The organism is urea is negative, whereas the proteus, which is of course the urea is negative and produce similar reaction to the triple sugar iron agar, its urea is positive. Identified by the group cytoglucation test into serogroups, that we can have the O group A, B, C, D, E, and E based on the antigen composition. Definitive serotyping of based on to the somatic antigen, flagellant antigen, virulent antigen is performed by the public health laboratories and epidemiology. It's not used for the diagnostic purpose. It is mostly for the epidemiological purposes. Salmonellosis in states is a notifiable disease. You will have to bring it to the notice of because it's a public health issue investigation to determine what is the source, and we should have to undertake that area in order to break the source of the infection. In our country, we don't have such rules and regulations for such diseases. The organism is difficult to determine the histological diagnosis in detected drive antibody rise and temperature. We can perform the Vidal test, which is based on to this intradermal injection of the antigen, and then to see it is an old area. It's a screening test. Now remember, Vidal test is not a diagnostic test for the Salmonella typhi. it is a screening test. We can have many false positive and false negative results. And even if you see someone has already, in the recent past, has a immunization against salmonella typhi, then you can have it. So without test is a screening test, and it's not a pathognomonic, it's not the definite diagnostic test. The treatments in the enterocolitis or diarrhea, it is self-limited, and we just need the fluid and the electrolyte replacements. Oral antimicrobial chemotherapy for the neonates or with person for the chronic diseases who have risk of developing septicemia or disseminated abscesses is required. The plasmid-mediated drug resistance is very common with this group, particularly with this salmonella typhi, and antibiotic sensitivity should be done if you are not getting a response in case of salmonella typhi. Treatment of child in case of enteric fevers and septicemia with metastatic infection is ceftriaxone or ciprofloxacin. Ampicillin in combination or alone and with that of ciprofloxacin for the chronic carrier of salmonella typhi for a longer period of time. And cholecystitis, sometimes you see you are having relapses, recurrent salmonella typhi disease. You are a main source of spread. You are a carrier. So there is no way left except that we'll have to go for the cholecystectomy. And this is sometimes becomes very important to abolish the chronic carrier state of the individual. The prevention is public health laboratories, personal hygiene, mere emphasis, sewage treatment, water chlorination, and also monitor the coliform count in the water so that it does not contaminate the drinking water. The stool culture from food handlers, hand washing, milk pasteurization, proper cooking of poultry, eggs, and meat is important to prevent this infection. There are two vaccines available against salmonella typhi. One is the capsular, virulent capsular polysaccharide, which is given intramuscularly, and the other one is the live attenuator, which is given orally. A new conjugate vaccine, which contains the capsular polysaccharide antigen coupled to that of the carrier proteins, it has also been used. It is safe. It is immunogenic in young children also. That was all about the salmonella and the rest of the escherichia. We have finished with all the enterics, and we have finished all the families of Enterobacteriaceae along with that of the other gram-negative rods. We will next time start with next topic. Thank you very much. Enjoy your time.